All right, welcome back, everyone. And today we are going to be looking at chapter 16, begins on page 314 in your text and page 99 in the course outline. And we've seen DNA is a nucleic acid made up of nucleotides. And we've talked about the fact that it replicates before a cell can divide. But what we haven't talked about is how it replicates. And that is what this chapter is all about. And I saw this image and I thought this was kind of hysterical, really. If you're a computer geek like me, and maybe you get this, maybe you don't. But if you remember anything about RNA versus DNA, you would get this. RNA single stranded, DNA double stranded. Hysterical. Anyway, the beginning of this chapter talks about how DNA was discovered and the different scientists that were given credit for the discovery of DNA. And rather than take a lot of time going through all of that, I'm going to show this video, which is one of a series that you can watch on YouTube if you like this, called The Secrets of Life. And they were really well done, but they begin with this just kind of a little bit of history on DNA, and then we'll get right into the structure of DNA and how it replicates.
50 years ago this spring, two scientists in England bolted together some brass parts and discovered the secret of life.
These were unlikely characters to be working on such an important problem.
The directions for a human being are written in code, 3 billion letters long. These instructions tell our bodies how to live, how to grow, how to die. Researchers call this code the sequence.
Welcome to Secrets of the Sequence. I'm Lucky Severson. Imagine you are tipping back a beer at an English pub when two rather wild-looking characters charge in and announce loudly that they have discovered the secret of life. What would you think? Monty Python? Time to water the whiskey to move on to a quieter pub? It's hard to imagine now what the pub patrons 50 years ago thought when two scientists, Francis Crick and James Watson, announced to the lunch crowd that they had made what is now considered the 20th century's most profound discovery. This year marks the Jubilee anniversary of the discovery of the double helix, the twisted ladder of DNA molecules at the heart of all life on Earth.
When Crick and Watson came bounding out that February day and went to their favorite pub, the Eagle, and they told people over drinks, "We've just found the secret of life," they weren't really exaggerating. It was the secret of life.
What started with Crick and Watson's excited bar talk at the Eagle Pub in Cambridge changed the world. Now we hardly blink when we see that DNA manipulation can make a mouse bald and make its offspring glow in the dark. DNA manipulation can allow plants to kill the pests that used to kill them. It can turn goat's milk into spider webs. It is now commonplace to see DNA evidence analyzed in crime investigations. Without an understanding of the double helix, we would not have had Dolly, the first sheep born without a father. And without the helix, Raelians could not have conceived their claims of the first human clone. The suit that Rael wears looks familiar. It calls up images from 1950 science fiction. By mid-century, the advances in the field of microbiology seemed to border on science fiction. In the post-World War II peacetime expansion, science was breaking loose from a narrow focus on military needs, the killing machines, and beginning to explore the basis of life. Electron microscopes were revealing the deep secrets of molecular structures in the nucleus of cells. The fact that deoxyribonucleic acid, DNA, carried genetic information was known, but few thought it was a central mechanism in heredity.
All the big players ended up getting things wrong. It was folks coming out of left field. The notion that DNA was the hereditary material itself, that was heretical.
So there were just a few people who felt that...
Well, through the 1920s and 30s, most smart money was on proteins. Proteins had all these different shapes and could clearly do so many things. They had to be the source of heredity.
Among the few contrarians were Francis Crick and James Watson. Both were working at the time on other research projects and not officially assigned to the study of DNA.
Crick and Watson working on the structure of DNA, these were hardly the orthodox scientific leaders. Jim had come from Indiana, where he was really trained as a zoologist, an interest in bird watching, and seemed like an unlikely character to be doing this at the age of 25. And Francis Crick, a retread physicist who had worked in the Admiralty in World War II, these were unlikely characters to be working on such an important problem.
The smart money was on Linus Pauling, a charismatic professor at Caltech in Pasadena, and the leading authority on the structure of protein molecules.
Well, in fact, Linus Pauling published first. He completely scooped Crick and Watson by publishing the structure of DNA before they did, except it turned out to be wrong.
Pauling theorized that DNA took the form of a triple helix radiating around a sugar phosphate central backbone.
And Crick and Watson knew right off the bat that that was nuts, because this sugar phosphate backbone is very negatively charged. You couldn't stick three negatively charged things in pointing at each other. They'd repel each other. How could Linus Pauling blow it like that?
Linus Pauling's error in basic chemistry gave Crick and Watson a break, a few more months in the race to create a theoretical model of the DNA structure. Key to their final success was a technology called X-ray crystallography. It could render the invisible world of molecules visible, but only to those few who could divine what the strange patterns meant. One of the best at using this extraordinarily difficult imaging science was Rosalind Franklin.
You can work out the shape of particular proteins and the shape of DNA, for example, and that's what Rosalind Franklin did. She took crystals of DNA, dried down DNA, shot X-rays into them, got photographic images, and those photographic images, to a practiced eye, like Francis Crick's, immediately told you that DNA was a helix. And that was...
It's a major discovery.
Oh, it's a major thing.
In a few days of intense work, Crick and Watson assembled the first rickety model of the double helix. The model was crucial to the two scientists because it allowed them to see and measure the three-dimensional relationship between the two chains of nucleotides, and thus determine if the theoretical structure would prove scientifically valid, and at the same time agree with what had been revealed in the X-ray patterns.
Rosalind Franklin's pictures of DNA, that is X-ray pictures, which revealed to the well-experienced eye of Francis Crick that DNA had to have a helical structure. Crick had been studying crystallography, and he recognized the helix when he saw it, was just an absolutely crucial part. But Crick and Watson, sitting alone without real data, would not have been able to figure out the structure of the double helix. And it's very clear that Rosalind Franklin, sitting alone looking at the structure of the X-ray crystal she had produced, didn't attempt to build models around it. So while science often seems like the work of the great individual, it is perhaps more than anything the work of an extraordinary community that both cooperates and competes at the same time.
In the half-century since, the 3D model of the double helix has been greatly refined and explored. Now, for the first time in experiments at the Molecular Biology Lab at the University of Geneva, the three-dimensional model has been expanded, adding a fourth dimension, time. The result? A fascinating image that reveals a dynamic living helix far more complex than a simple string of code.
It's a tremendously important, tremendously exciting chase about things, secrets of the world, and you want to know them first. It's a chill. I think most people have never had the experience of being the only person in the world to know some fact about nature.
So the key to our genes may lie not in where they sit, but in how they dance. Biology's complexity seems to grow exponentially as science digs deeper. It seems the more we learn, the more there is to learn. And it poses a question whether it will ever be possible to know it all. That, too, is a secret of life.
The Secrets of the Sequence teaching material.
So there were several people involved in the discovery of DNA. Watson and Crick usually get most of the credit for that. Francis Crick actually died in 2004, but he was at least able to see the 50-year anniversary of the discovery of DNA in 2003. So that was great. And as far as I know, James Watson is still alive. I believe he's about 92 years old. Then there were some others, Maurice Wilkins and Rosalind Franklin, that also, as that video pointed out, were involved in figuring out this DNA structure. So Watson and Crick, although they get the majority of credit for this, they really couldn't have done it without Rosalind Franklin using that X-ray diffraction technique to figure out that DNA was a double helix. So when you think about somebody like James Watson, who is still alive, in his lifetime, not only did he see the discovery of DNA almost 70 years ago, but he was able to see the conclusion of the Human Genome Project, which catalogs the entire human genome, which consists of something like 3 billion letters. He has been able to see all kinds of advances in DNA technology, which includes everything from cutting and pasting genes into bacteria to cloning animals and even making designer babies. So that's kind of what this section is going to be about. We're going to start with just a look at how DNA is replicated so that cells can divide, and then we'll look at DNA technology and some of the latest and greatest things that we are now able to do because of this discovery. So concept 16.1 is on page 99 of your course outline and talks about, again, the pattern in that photo suggested that the DNA molecule was made up of 2 strands forming a double helix. That's kind of like a twisted ladder. So what is DNA and what do we need it for? This clip will talk about the 4 functions of DNA, also listed on page 99.
To fly a rocket ship, you need to be an optimist. No astronaut launches.
One of the functions DNA has to perform is replicating itself. There is a lot of replicating and reproduction going on in the living world. Plants and animals creating offspring, their cells dividing, and unicellular protists and bacteria dividing too. Each and every cell in every organism has to have a copy or copies of the DNA unique to its particular species. While it's important for DNA to replicate accurately so that when cells divide, each receives exactly the same genetic information as the other, it is just as important, in terms of creating the Earth's incredible living diversity, that an error or mutation occur occasionally, and that these mutations can be transmitted to future generations, which is just what happens when a mutation occurs in the DNA of a sex cell. The diversity of life here on Earth is largely a result of mutations that have occurred in strands of DNA over the last 3 billion years. Another requirement of DNA as a genetic material is that it be able to store the information that determines the characteristics of organisms and the cells of which they are made up. On the surface, this sounds like an easy and obvious goal, but there are animals, like certain species of turtles, that can live hundreds of years, and plants like redwoods that can live thousands. That is a long time to store information in a potentially volatile chemical environment. If the cells in an organism started losing parts of their biochemical memories on a mass scale and forgetting which critical cellular functions to carry out, it wouldn't be long until the organism died as a result of this cellular memory loss. Finally, DNA or any other potential genetic material has to be able to use the information it stores to direct the synthesis of the structural proteins and enzymes necessary for the operation of the organism and its cells. Structural proteins define and maintain the shape of a cell or organism, while enzymes are large proteins that are critical to carrying out all the various chemical reactions essential to life. The elegant simplicity of DNA is at the heart of its performance of its varied tasks as the molecule of heredity. Each sequence of bases spells out, in a sort of biological Morse code, a unique set of genetic instructions. In a length of DNA only 10 nucleotide pairs long, the 4 bases can exist in over 1 million different combinations. As the average chromosome of higher organisms is billions of nucleotides or base pairs long, we can see that the information storage potential of a strand of DNA is tremendous. But the huge number of base pairs in the DNA of any given chromosome would also appear to make the potential for replication errors tremendously high.
Okay, so once again, looking at the 4 functions of DNA. So one function, and that's the first function that we're looking at in this chapter, is how DNA replicates so that cells can divide. Also, in the past chapters, we've talked about mutations, which are spontaneous changes in DNA, and those are also important for the diversity of life. A lot of times people think mutations are detrimental or bad, and they can be, but they also do provide genetic variability, which is a good thing. Then we have DNA provides storage of information to help us continue the species, and finally a process known as translation, which we're going to look at in the next chapter, which is a process of converting DNA into proteins, or pretty much our genes into those traits that we've talked about in genetics. So we're going to begin with a look at DNA structure, and a couple things hopefully you already remember about the structure of DNA is that it is a nucleic acid that is made up of these nucleotides. So this is the unit molecule that makes up the DNA or nucleic acid. Remember the Na in DNA or RNA stands for nucleic acid. And we have 3 basic parts here. We have this 5-carbon sugar, which is in DNA is deoxyribose, in RNA it's ribose. We have this highly energetic phosphate bond. So we've talked about phosphate is usually shown here in yellow, depicting that it is a highly energetic molecule. Something like ATP, adenosine triphosphate, has 3 phosphates, and that's our energy-carrying molecule in the body. And so we have that. And then the third part is this base over here. And the base is going to vary. There's really only 4 bases in DNA. You may have learned them before, A, T, G, and C. A is for adenine, T is for thymine, G is for guanine, and C is for cytosine. So we'll talk about those bases. And that really is the letters, or are the letters of the alphabet of life. Now, another important thing to note, anytime you look at organic molecules, and remember this is a hydrocarbon, so it's organic, is numbering these carbon atoms so you know what's attached to what. And so when we start out with this 5-carbon sugar, on the first carbon, we're going to call this carbon number 1 wherever the base is attached, then carbon number 2, then carbon number 3 has this OH group, carbon number 4, and carbon number 5 has the phosphate group. This will become important when we start talking about the direction or how these nucleotides pair up. And so when you see this little prime symbol here, so we would call this the 1', 2', 3', 4', or 5' carbon as we go along. Now, when 2 nucleotides bond, they form this special bond where the 5-carbon attaches to a 3-carbon, and this is called a phosphodiester bond. Notice the 5' end is up, the phosphate end is up, and down here the OH group is free. So again, you're going to hear these referred to when we get to the direction that the bonding occurs. So a look then at the structure of DNA. Here's the outside of the ladder. Remember, it's a double helix, so it's almost like a twisted ladder. So we've got the two sides of the ladder, and the side includes the sugar and the phosphates. And then the rungs of the ladder are in the middle, and that's where we see these bases. So again, we have these 4 bases, usually represented with the letters A, T, G, and C. And you can remember that. Somebody told me, a student said, remember at the golf course, if you like mnemonics. So anyway, and again, remember those carbon numberings down here so that we see that these bases are attached to the first carbon. You've got that OH group on carbon number 3, phosphate group on carbon number 5. So we have the 5' end up here and the 3' end open down at the bottom. Now I say top and bottom, but keep in mind this is in a 3-dimensional space, so there's really no top and bottom, just one end or another, which is why we refer to those as the carbons. What carbon is open and what direction they're going. Again, I'll get to that in just a second. But why is it that A pairs with T and G with C? And the reason for that has to do with hydrogen bonds. So you can see here that A and T form 2 hydrogen bonds, whereas G and C form 1, 2, 3. Now remember, we represent hydrogen bonds as dotted lines because these are weak bonds, very easy to break. They're also bonds between molecules. So we've got 2 hydrogen bonds between A and T and 3 between G and C. And so it's their molecular structure that makes them want to bond this way. So you can remember A always bonds with T, these are the straight letters, and G with C, these are the curved letters. That's one way to remember that. So here's our twisted ladder, and if we untwist it, and again look at the chemical structure of a piece of DNA, we can see here's A with T, G with C, C with G, A with T. Doesn't matter which one's on which side as long as the A pairs up with T and G with C. And then the outer rungs, remember, are going to be the sugar and the phosphate groups. Now here's another thing to notice. It's almost upside down on one side compared to the other. So here we have the 5' end up and the 3' end open on this side, and it's just the reverse on this side. So we call this anti-parallel. That is going in 2 different directions, and that is going to be important too as we see how DNA replicates here in a minute. So we also have this term semi-conservative model. This is the bottom of page 99. And Watson and Crick's semi-conservative model of replication predicts that when a double helix replicates, each daughter molecule will have 1 old strand, that is, it's conserved, and 1 newly made strand. So in dark blue here, as you follow this, here's the original parent molecule. And what will happen is enzymes will come along, unzip those hydrogen bonds, separating the strands and exposing the free bases. Now in the nucleoplasm, because of the food that you eat that you have digested down to unit molecules, you're going to have some free nucleotides swimming around or floating around there. And so the A will attract a new T and the C a new G and so forth on both sides. And that's essentially how DNA is replicated. So the daughter DNA molecule consists of 1 parental strand shown here in dark blue and 1 new strand shown here in light blue. Again, that term semi-conservative, semi meaning half of the parental strand is conserved in dark blue and the other half is new in the light blue. Now on to concept 16.2, page 100 in your outline, looking at a closer look at DNA replication. And since the 2 strands of DNA are complementary, here's a new term. So think of this word complementary. It doesn't mean they're identical, but A with T and G with C. In other words, A is complementary to T and G is complementary to C. It's like a knife and a fork. They're not the same thing, but they work together. And so we do say that the 2 strands of DNA are complementary. Each strand acts as a template for building a new strand in replication. That word template means a pattern for assembly of something else. In this case, it's providing a pattern for assembly of a new strand of DNA. In DNA replication, then the parent molecule unwinds and 2 new daughter strands are built. Again, that doesn't mean they're female. That's just the term used to describe the newly built strands. And they are going to, again, follow those base pair rules. So when we get started, in order for this to begin, we start at particular sites called the origins of replication. This is where the two DNA strands are separated, and they open up what is known as a replication bubble. In eukaryotic chromosomes like our cells, we may have hundreds or even thousands of origins of replication, but in prokaryotic cells, because they have much less DNA, and remember prokaryotic before the nucleus, right? Those are our bacterial cells. In that case, they only have just the one origin. Replication proceeds in both directions from each origin until the entire molecule is copied. So it is what we call bidirectional. So we have done most of our studies on DNA replication by looking at prokaryotic cells like bacteria. In this example, we're looking at E. coli, Escherichia coli, which is a common bacteria that inhabits our colon, our digestive system. And when you look at it, remember prokaryotic cells have a single circular piece of DNA. There's their chromosome. And so enzymes are going to attach to that parental strand of DNA. Again, keeping with the color code, dark blue is the parental, light blue is a new strand. And what will happen is also enzymes will start attaching the correct nucleotide bases. A would attract T, G with C, and it will continue, notice, in both directions, bidirectional, and it continues and continues until we end up with two new daughter strands that are semi-conservative, right? Half old, half new. This is an actual micrograph that was taken where you can see these origins and how the DNA is replicating. But remember, in eukaryotic cells, because there's so much more DNA involved, and this has to happen very, very quickly. Anytime, in order for a cell to divide, we have to first replicate the DNA. So this has to happen very quickly. And so we have many origins, as I mentioned before. You can even have thousands all working simultaneously to unzip that original parental strand and get it copied very quickly. So here is what we call the replication bubble. Notice that we are going in both directions, bidirectional, so that we can replicate this very, very quickly in order to get our two identical daughter DNA molecules. So how does this work? Let's take, again, even a more closer look. And here we're looking at some of the proteins involved in initiation of DNA replication. So it all begins with some enzymes attaching to that parental strand of DNA, again shown in dark blue. Here we have topoisomerase, here we have primase, here we have helicase. That should look familiar. What are these proteins? They all end in ASE, so everybody, hopefully you know what they are, right? These are enzymes, and they are named for what they are doing. Now, topoisomerase is a type of enzyme that corrects overwinding ahead of replication forks by breaking, swiveling, and rejoining DNA strands. So this is kind of leading the way. This is the replication fork wherever it's unzipping. And so topoisomerase is controlling either over or under winding in the forefront here. And then helicase, as the name implies, is working at the helix right here. Helicase is an enzyme that untwists the double helix at the replication forks. And then primase is a special enzyme that can start an RNA chain from scratch and add RNA primers. What are RNA primers? Well, that word primer means to prepare something, and that's exactly what it does. This primer will prepare this reaction to bring up the complementary bases. So it starts originally with a piece of RNA, but that RNA is going to be replaced by DNA later. And then we have some other proteins involved you can see here we'll get to in a minute. Now, another important group of enzymes are a group of enzymes known as the DNA polymerases. And as the name implies, they produce DNA. And so there are several different kinds. We'll just call them the polymerases or polymerase. and they are responsible for bringing up these complementary bases. So these polymerases catalyze the elongation of a new DNA at a replication fork. So initiation, just let me back up a minute. Initiation here is when all of these enzymes load up, and then elongation is when we start making the new strand. And so we are bringing up the complementary bases, A with T, G with C, as it continues on, and we get, again, that new strand being prepared. Most DNA polymerases, again, require that primer and a DNA template strand. So this would be the template, so it provides pattern for assembling the new strand. So the T would attract A, G with C, and so forth. Then we have what is known as antiparallel elongation on page 101. Remember, this is going in two different directions. And so the antiparallel structure of the double helix affects how replication is going to occur on either end. And as it mentions here, DNA polymerases add nucleotides only to the free 3' end of a growing strand. Therefore, a new DNA strand can elongate only in the 5' to 3' direction. You'll come across this quite a bit in your readings. And as you continue on, if you take anatomy and physiology or microbiology, you study this again and again, you'll hear this reference all the time, this direction, 5' to 3'. But since we have this antiparallel elongation along one template strand of DNA, the DNA polymerase synthesizes a leading strand that is continuously moving towards a replication fork. So we see here, if you're going towards the replication fork, and we start with the primase and the primer, as you see here, and we bring up the complementary bases one right after another. This is called continuous assembly, and this is what we also refer to as the leading strand. There's no gaps. And this is because it's going towards the fork. But as we see to elongate in the opposite direction, it's going to pose a little bit of a problem. And so in this case, we're producing the lagging strand. It gets its name because DNA polymerase must work in the direction away from the replication fork, and this causes it to kind of twist in a unique way and leave some gaps. And so we end up with a series of segments called Okazaki fragments, which are then later joined together by another enzyme known as DNA ligase. So here's a clip that'll kind of put all this together, and then we'll talk about what DNA ligase does as well.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Okay, so again, an overview of the synthesis of the lagging strand compared to the leading strand. So we can see here the lagging strand is going away from the fork, and as it does that, it forms these little sections, these fragments, which are those Okazaki fragments, whereas the leading strand forms continuously towards the fork. Overall, the directions of replication are bidirectional, going both ways. So the lagging strand, again, away from the fork, the leading strand towards the fork, the lagging strand discontinuous, the leading strand continuous. And so what happens then is we've looked at some of these enzymes, but we need some other enzymes to help us handle this discontinuous situation on the lagging strand. And because there are gaps, we now have a new enzyme to add to this mix, which is DNA ligase. And this is a really important enzyme. We're going to talk about this later because when we get into DNA technology and we cut and paste genes, we use this enzyme as the DNA paste. So many of these enzymes will cut, but ligase is our paste. And what it does is it pastes in these sections so that it will end up being continuous assembly when it is finished. But again, the lagging strand is the only strand that has DNA ligase as part of it. So we've seen all of these other enzymes working, and now we're adding DNA ligase as well. So in concept 16.3, talking about a chromosome and how it consists of a DNA molecule packaged together with proteins. Some of these we've already talked about, this term chromatin. Remember that most of the time the DNA is all unwound in the cell nucleus in the form of chromatin. But when the cell is ready to divide, that's when it condenses and forms chromosomes. So how do we get all of this chromatin? And I've given you this analogy before, that DNA is a long fibrous molecule, and it's kind of like taking DNA or taking thread off a spool. If you've ever done that before, you'll know you'll have a giant pile of thread. How do you get all that thread organized? By wrapping it tightly around the spool. That's exactly what happens with DNA. How do we get all that DNA into teeny tiny cells? It's through this special DNA packaging, which is wrapping the long fibrous DNA around proteins, just like thread around a spool. So here's how this works.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Okay, so once again, what we see are these series of proteins that are like the spools shown here in purple, with the blue is the long fibrous DNA molecule. So again, as we see, there's many, many different kinds of loops as they loop around these histones, these proteins, and then they form a double loop around 4 of those histones. Collectively, that is known as a nucleosome. So then these nucleosomes, or beads on the string, start again condensing into these chromosomes that we actually see then under the microscope when we're looking at cells that are in the process of dividing, which we have seen, at least most of you have seen, those of you who went to that lab. So when all of this DNA is being replicated, as I mentioned, A is supposed to pair with T and G with C, but sometimes there are mistakes. And so fortunately, we do have some extra enzymes that are involved in going through and proofreading the newly made DNA and replacing any incorrect nucleotides, which is really good because we've talked about genetic disorders, things like sickle cell anemia, which is caused by just one mismatched pair, which then causes the hemoglobin to be produced faulty, which then affects the red blood cell, which then affects every system of the body. So one little mistake here can cause a lot of problems. But fortunately, as I said, we do have these repair enzymes that correct errors in base pairing. What kinds of things can damage DNA and cause these changes? A lot of stuff, but exposure to harmful chemicals or physical agents, especially I think everyone knows cigarette smoke is one of the a huge carcinogen that causes a lot of different kinds of mutations. And X-rays, we don't want to have too much exposure to any kind of radiation. So in nucleotide excision repair, a nuclease, what is that? Nuclease ends in ASE, right? So it has to be an enzyme, cuts out and replaces damaged stretches of DNA. So now we have a new enzyme called nuclease. And so we have really 3 enzymes involved in repairing any of these mismatched pairs. So we have the excision repair through the nuclease, which will cut at the points that need to be replaced. DNA polymerase will bring in the correct base pairs, and then ligase will paste them in. So nuclease is kind of the DNA scissors, and DNA ligase is the DNA paste. And that's how we fix, hopefully, most of the problems. Not all of them, unfortunately, but most of them. What is the evolutionary significance of altered DNA nucleotides? Well, as I mentioned before, the error rate of proofreading repair, it's low, but it's not 0. So sequence changes may become permanent and can be passed on to the next generation, and we call these mutations. Are these good or bad? We've talked about this before. You know, a lot of times you hear the word mutation and you think it's going to cause some gross abnormality. It could, but many times these provide new genetic information that may actually be better from an evolutionary standpoint. So the theory of natural selection here talks about that those that have the best genetic components or best genes survive and pass that DNA on to the next generation. So it really depends on the environment whether these mutations are advantageous or detrimental or even deleterious, which means it could cause death of those that inherit them. So let's do a little try this. This is on the bottom of page 101. And see if you remember which enzyme does what. So which enzyme opens and unwinds the DNA double helix? Here's the keyword there, helix. Remember our enzymes end in ASE, so this one's helicase. Which one synthesizes RNA primers? Hopefully, again, keyword here, primase. The next one assembles new complementary strands. This one is the group, as I mentioned, that are polymerases, but you have to be careful if they're making new strands of DNA from DNA. In other words, DNA is replicating. it's DNA polymerase, because in the next chapter, we're going to see how DNA is converted to RNA, and then we use a different group of enzymes known as RNA polymerases. Which one joins discontinuous segments? This one is only associated with the discontinuous side, the one that has the Okazaki fragments, and needs to glue those together. So this would be the DNA glue ligase. And here's our DNA scissors that cut out damaged stretches of DNA. That would be the nucleases. Over to page 102, talking about this last section of replicating the ends of DNA molecules. And it mentions there that limitations of DNA polymerase create problems for the linear DNA of eukaryotic chromosomes, and the usual replication machinery provides no way to complete the 5' ends. So repeated rounds of replication produce shorter DNA molecules with uneven ends, and this could be very detrimental. But fortunately, eukaryotic chromosomal DNA molecules have special nucleotide sequences at their end called telomeres, and they work with an enzyme called telomerase that catalyze the lengthening of these telomeres in germ cells. Remember germ cells? Those are the ones in the male and female gonads, which would be the male testes and female ovaries that are involved in replication for sexual reproduction, that is, meiosis. So telomerase then catalyzes the lengthening of telomeres in the chromosomes that are in those germ cells, and then those will be passed on, and that will help then conserve the DNA so we don't lose it as it continues to get shorter and shorter. So here's a clip on how that works.
The ends of linear chromosomes pose unique problems during DNA replication. Because DNA polymerases can only elongate from a free 3' hydroxyl group, the replication machinery builds the lagging strand by a backstitching mechanism. RNA primers provide 3' hydroxyl groups at regular intervals along the lagging strand template. Whereas the leading strand elongates continuously in the 5' to 3' direction all the way to the end of the template, the lagging strand stops short of the end. Even if a final RNA primer were built at the very end of the chromosome, the lagging strand still would not be complete. The final primer would provide a 3' hydroxyl group to synthesize DNA, but the primers would later need to be removed. The 3' hydroxyl groups on adjacent DNA fragments provide starting places for replacing the RNA with DNA. However, at the end of the chromosome, there is no 3' hydroxyl group available to prime DNA synthesis. Because of this inability to replicate the ends, chromosomes would progressively shorten during each replication cycle. This end replication problem is solved by the enzyme telomerase. The ends of chromosomes contain a G-rich series of repeats, called a telomere. Telomerase recognizes the tip of an existing repeat sequence. Using an RNA template within the enzyme, telomerase elongates the parental strand in the 5' to 3' direction and adds additional repeats as it moves down the parental strand. The lagging strand is then completed by DNA polymerase alpha, which carries a DNA primase as one of its subunits. In this way, the original information at the ends of linear chromosomes is completely copied in the new DNA.
So, as it mentions here, it has been proposed that damage to telomeres is connected to aging. So, with that in mind, there's been all kinds of research looking for the fountain of youth. What can we do to prevent our telomeres from fraying? And that, in turn, hopefully would prevent us from aging. So, here's a clip on that.
Good to see you.
Yeah, hey, what's with the polka dots?
What do you mean?
Polka dots are out. It's polka.
While many search for the proverbial fountain of youth, you might be wondering, why do we age in the first place? What is it about our bodies or cells biologically that causes us to grow old? There's a variety of internal and external factors such as diet, exercise, or environmental stress which all contribute to cell damage and repair and affect the rate of aging. But the surprising truth is that, apart from these, we actually have a biological clock buried within our genetic makeup, and this clock can only run for so long. In other words, we're programmed to die. Your body is made up of trillions of cells which are constantly going through cell division, and every time they divide, they make a copy of their DNA as well. This DNA is tightly packed into structures called chromosomes, of which humans have 23 pairs. The problem is, DNA replication isn't quite perfect and skips over the end of each chromosome. To protect against important DNA information being cut out, we have something called telomeres on the end of chromosomes, which are essentially meaningless repeats of DNA that we can afford to lose. But every time our cells divide, these telomeres become shorter and shorter until eventually they've been entirely stripped away, at which point the cell no longer divides. Some flatworms are able to endlessly regenerate their telomeres, making them effectively biologically immortal. But their lifespans do vary, and they're still susceptible to disease, further suggesting that aging is a mix of genetic and environmental factors. But why don't our cells do this? Ultimately, this replication limit actually helps to prevent cancer, which is the uncontrollable growth of cells and evasion of cell death. The point at which a cell stops replicating is known as cellular senescence. In humans, this replication limit is around 50 times. Once it's reached, the cell gradually begins to lose its function and die, causing age-related characteristics. This also helps to explain why life expectancy is a strongly heritable trait from your parents, because you got your initial telomere length from them. Got a burning question you want answered? Ask it in the comments or on Facebook and Twitter.
So the big question is, do we want our telomeres to shorten or not? Because, as it mentions on your outline, so it's been proposed that damage to telomeres is connected to aging, but as it mentioned on that clip, the shortening of telomeres might protect cells from cancerous growth by limiting the number of cell divisions. And there's evidence of telomerase activity in cancer cells, which may allow cancer cells to persist. So some research suggests that dietary supplements might offer a means to lengthen telomeres. And again, if you take these supplements, maybe you could live longer. But then there's also evidence that says taking things like telomerase may actually be cancer causing. So it is a dilemma, and there is no real good evidence on either side of what's going on there. So in the meantime, always err on the side of caution. I would probably stay away from taking those supplements, and not only that, but they're super expensive. If you're looking for the fountain of youth, maybe do some of those other things that keep us young, like exercise and eat right. So with that, that pretty much wraps up the chapter 16 on DNA replication. And next time we will be looking into how DNA is converted to proteins in chapter 17. And I will see you then. Have a great day. Be well.